Okay, everyone, we're going to go ahead and get started. Thank you for coming to our TRI Innovation Seminar Series for the month of March. We are welcoming our cohort of our 2017 KL2 Scholars to talk about the research they did for TRI, share some of the innovation they are bringing to the scene. So without any further ado, I'm going to stop sharing my screen here in just a second and let Dr. Nabawisi, who was our first presenter, share her screen. Okay, and I'll turn it over to Dr. Nabawisi.
Okay, good afternoon, everyone. I would like to thank TRI for this opportunity to share my work and the support they provided, 2-3 years ago now. So I am Rosemary Nabawisi, for those of you who do not know me. I am an assistant professor in the Department of Pediatrics, and I conduct health services research in addition to using implementation research and community-based engagement to address health disparities in infant mortality, specifically caused by childhood injuries. I also serve as the Senior Director for Research and Evaluation in the Division of Diversity, Equity, and Inclusion. So I am presenting to you work that started out as a KL2 project, and it was in collaboration with the Birthing Project, and we looked at educating African American mothers in Southeast Arkansas. So of course, TRI supported this work. And after my talk, you'll be able to describe the prevalence for infant mortality. You'll understand what sudden unexpected infant death is and how it presents in disparities. And then you'll also be able to understand the barriers and facilitators that communities in rural Arkansas face. So looking at the infant mortality rate is the number of children under 1 year of age that die per 1,000 live births. So looking back about 15 years ago, our state rank was 37th, right? And we were pretty close to the national infant mortality rate. Come down to the most recent data from the CDC, the national infant mortality rate has gone down, but ours has, you could argue that it's just rotating around the same area and we're not really making improvements. And right now in 2018, we were ahead of just Mississippi and Louisiana. So infant mortality is a big problem still in our state. And outside of prematurity and congenital anomalies, sudden unexpected infant death is the cause of, is the major contributor to infant mortality. And sudden unexpected infant death, like the acronym is, it's the unexpected death of a child who's under 1 year of age. And the contributing factors, about close to a third, the causes of death are unknown, but the other quarter of the deaths are due to accidental suffocation and strangulation in bed. And then the rest are due to sudden infant death syndrome. And sudden infant death syndrome is SUD that cannot be explained after conducting a death scene investigation, an autopsy, or a clinical history. And so why are we so concerned about SUD, which is sudden unexpected infant death? We're still concerned about it because of the disparities, the racial disparities, and then the geographical disparities. So you find that twice as many African-American infants die from SUD compared to white infants. And then you see the ratio there, rural to urban. And that's why I did this work in Chico County. And then some of the other contributing factors are the evidence-based safe sleep interventions are not uniformly available in the rural communities. And then accordingly, that follows with adoption is not I guess it's not uniformly practiced, right? The safe sleep recommendations. And so, like I said before, you have those disparate practices. Granted that the white infants or white parents are not practicing the safe sleep recommendations at close to 100% rate, right? So we have still a gap of 30% among the white parents, but then significantly much higher among African-American parents. So of course, we know Chico County is one of the red counties, and it's really underserved, as you can tell from the high federal poverty level and the significant infant mortality rate. So the research gap that existed was, you found that when parents come in contact with the healthcare system, only close to half of them get correct advice. So safe sleep guidance that's given to them when they come for their well visits, right, with their children. Close to 20% get incorrect information. And then a quarter of them do not get any anticipatory guidance about safe sleep. And yet, vital signs from the CDC indicate that caregivers who received correct advice were less likely to basically not follow the AAP guidelines. So they were less likely to put their infants on their stomach for sleep. And I'll go over what the recommendations are for safe sleep. So safe sleep educational interventions can be delivered in various forms. Most commonly, they're delivered in hospital settings, typically when the mother has delivered the infant. But the argument is that those circumstances are very stressful for the mother. You don't expect them to retain as much information after they've had their baby. So the argument is to provide the safe sleep education in a community setting or in prenatal context, right? So these are the various implementation strategies that are used to deliver the education. Safety baby showers are usually delivered in community settings, although also here in the NICU and in the resource center here at SCH, we do deliver safety baby showers. So the ABCs of safe sleep include the American Academy of Pediatrics recommends that every infant should sleep on their separate sleep space by themselves. They shouldn't be sleeping with pets, shouldn't be sleeping with stuffed animals, they shouldn't be sleeping with adults or siblings. They should be put to sleep on their back, and the recommended sleep surfaces are a crib, a bassinet, or a play yard. So those are the level A recommendations. There are several other recommendations, but we concentrated on the ABCs. And so the table I have here in front of you is a study that was done at Johns Hopkins that showed the use of a flip chart in an outpatient clinic. They had a health educator walking the parents through this flip chart and educating them about safe sleep. And they were able to show that the knowledge scores of the parents who were in the intervention were much higher than those who were just in a standard group where they get routine anticipatory guidance without any emphasis on safe sleep for infants. And then you can also see the comparisons there on specific survey questions that they administered. So parents who were in the intervention group were less likely to agree with co-sleeping, and then they were also more likely to refute that discomfort happens to the baby if the baby is sleeping alone. So these are some of the attitudes that parents have to justify co-sleeping. And so we use this evidence to deliver the safety baby shower, because using a flip chart and using the safety baby shower is sort of relatively the same in administering safe sleep education. So what is a safety baby shower? So the safety baby shower was developed following the traditional baby showers. So you have parents and their significant others show up to party atmosphere where where we're celebrating their expected baby. And so instead of giving out random gifts, we give gifts that are safe sleep and home safety focused. And so the core components of the safety baby shower are the safe sleep education. We also educate them about distress crying so that we avoid head injuries or shaken baby syndrome. And then we also give them education about the child passenger safety and home safety. Modifiable aspects of the baby shower are who delivers it, right? We could train community advisors, we could have health educators deliver the education. Games are part of this shower, and people can dictate what sort of games they integrate into the baby shower. And then we provide them food, because the expectant mothers, you want them to be comfortable and happy. And so the delivery includes a didactic learning. They do have hands-on training. We set up a play yard, and we have blankets in there, stuffed animals, and then after the didactic learning, we ask the parents to set up an ideal sleep space for their infant. So they go in and they have hands-on training with that. And then there's also a car seat, and they learn how to strap a doll in the car seat. And then we also give them reinforcement gifts. We have t-shirts that say this side up for the baby and what have you. So our specific aims, we had two. We explored the rural African American parents' perspectives, as well as their community advisors, their perspectives on the safety baby shower. How acceptable was it to them, how feasible it was in their community and in their context. And we were looking for suggestions of how to adapt it so it could be readily adopted in their context. And the second aim was to tailor the safety baby shower so that it could fit within their community. And we used the implementation strategies they suggested. So our approach was to conduct a qualitative study through an academic community partnership with the Birthing Project, like I mentioned earlier. And the Birthing Project is a nationwide community organization whose mission is to improve birthing outcomes for African American parents. And their goal is mainly to connect usually young mothers to resources in their communities. And we felt that their mission aligned with the specific aims of this project. So I set out to do primary data collection. Our inclusion criteria were community advisors and parents who were members of the Birthing Project, and they had to be older than 18 years of age. So we conducted 5 focus groups. We interviewed the parents separate from the community advisors, and we had 3 key informant interviews as well. We used the consolidated framework for implementation research guide to develop our interview guide. We recorded the interviews audially and then took field notes. So that data was transcribed, and we analyzed the data both manually as well as with the MaxQDA software. We used the CIFA framework to generate codes before looking at the data. And then we also came up with some new codes and themes inductively while we're looking at the data. So altogether, we had 15 community advisors and 6 parents. I'll say we collected the data over a period of, I'll say 3 months or something. It was during the spring. And I remember one time we drove down to Lake Village, and there was only one mother with like 5 children. So instead of a focus group, it became sort of a key informant interview. We had more interviewers than the interviewee. But the mean age of our community advisors was 54, and the expectant parents were half their age. So these are some quotes that we got when we asked the community advisors how they used to put their babies to sleep. So these are usually older women, and so this is what they used to do. Okay, some of them would put their babies on their chest. They would lay them on their side. Granted, back then the AAP had a recommendation that putting your baby to sleep on the side was fine too. So during the focus groups, they did say, the community advisors said that they had learned, and now they were teaching their children as well as their little sisters in the bathing project, the AAP recommended guidelines. So some exemplary quotes I pulled up here. You can see the bolded one. This one, I believe this was one of the parents, and they were talking about the fact that Chico County had lost their obstetrician and they did not have a labor and delivery at the main hospital. So they had to go out of state for care when they delivered their baby. So she said, they not only just teach you how to take care of your baby, but just taking care of yourself once you have the baby. She was referring to this is what they used to do when they still had OBGYN in their county. And so they were complaining about the care they were getting from Mississippi. So the identified barriers and facilitators we found from analyzing the qualitative data span this spectrum. So of course, most of them complained about they did not have transportation. The safety baby shower was being delivered in Lake Village at the health department, but Chico County has two other towns, Eudora and Damot. So the people from the other towns felt that they did not have the resources to travel to Lake Village to participate in the safety baby showers. And these were being held monthly on the first Thursday of every month. And so in addition to lacking childcare, most of them were like, we have to stay home with our other children, so we cannot pick up and come to the safety baby shower. They did not have enough social support. They also said the showers took place during the time when some of them were working. So they were asking for flexibility in time. And then again, like I said, they needed an option to decentralize the location for the intervention so that people from the other communities could get to it as well. And then the theme that came out from the inductive data analysis was their cultural norms and their beliefs. They felt that co-sleeping was bonding. They were protecting their infants. Their babies slept longer when they were on the bellies or sleeping with them. And then they also needed some education about the baby choking while they're lying on their back. That's what they think, but actually they are more protected if they're lying on their back from choking. And then the other big thing was a lack of a bathing hospital in the county. So these are the summary results. We had the subcodes that ended up generating the main themes of attitude, social influences, and then trust. Trust was another one that came out of our inductive analysis. So they were like, we have to trust the agency that's giving us the information. We have to be able to trust the interventionist, whoever's delivering the safety baby shower. And then back to the quote I read, they mentioned that, the parents mentioned that you should not just be caring about my infant, you should also care about the health of the mother, because if the mother is not well, how will the mother be able to take care of the infant? So from those results, we conducted our second aim. I think, let me go to the next slide. The second aim was the evidence-based quality improvement process, whereby we had monthly meetings between the community partner leadership and then the academicians. So we had the intervention experts, we had myself, public health researchers, and we sat down and looked at the summary results and the suggested implementation strategies and decided on incorporating the intervention into a personal shower, because both the parents and community advisors recommended that they would be willing to give us 30 minutes in their personal showers to give this safe sleep education. And so we felt that this was a great catchment audience. So you had an expectant mother with her family and friends. They had the festive atmosphere already, and we could tap into one of the community advisors perhaps to present the safe sleep education. And so everyone would be abreast of the proper way to raise the child. And so we decided on developing the personalized safety baby shower, that was our next step. We had to put in some applications to get funding to do this work, and that happened right before the pandemic started. This is just summarizing the barriers in the CIFA framework, the Consolidated Framework for Implementation Research. They talked about the safety baby shower being significantly complex, and it was costly because if community members are to implement the shower, they have to do what to pay for the reinforcement gifts. And then, of course, all the resources and the household environment and what have you fit into these domains for the framework. So basically, we concluded that identifying transformative implementation strategies that are developed by community-informed processes would support improved safety baby shower delivery and adoption and result in sustainable improvements in rural underserved communities. And actually from this work, I was able to put in an application to Robert Wood Johnson Foundation that was looking at the social and built environment for these communities, because these are the factors that actually affect communities and individuals adopting public health recommendations. And I was successful in getting that grant. So in a couple of years, we'll be developing a bold strategic initiative to improve the culture of health in Chico County. And that's all I had.
Okay, I'm not seeing any questions. So we can move right along to Dr. Shinky.
Thank you for attending today's presentation. It's on my KL2 project, the role of PHF19 as a promoter of tumorigenicity and therapeutic target in multiple myeloma. It's going to be a little bit technical. I try to keep it simple. If you have any questions, you can just interrupt me, please. So what is multiple myeloma? It's a cancerous disease of plasma cells. So every healthy person has plasma cells, and then if these become abnormal and clone and start growing, they can eventually become myeloma cells. Myeloma is the most common malignancy of the bone marrow, roughly 30,000 cases a year in the US. You can see here that the age distribution is such that it's usually a cancer of the more elderly, but we do see also patients in their younger years. And the problem with multiple myeloma is that it's really not curable for the majority of patients. They can live long, and with newer treatments, they can live 10 years or beyond, but eventually still most patients will die. And so that's really a problem for patients that are diagnosed in their younger years. So here the overall survival, you can see it has improved due to better medicines over the last decade. But again, at this time point, 50% of patients probably were dead after 5 years. Again, that's a little bit better by now, but still there's a lot we can improve in terms of outcomes. So obviously everybody's trying, or everybody in myeloma research is trying to look for how to cure this disease and what approaches would be the best. So multiple myeloma has also different clinical stages. There are precursor stages called MGUS and smoldering, which are actually not cancerous yet. The cells just sit in the bone marrow but don't cause any problem. So we don't treat that. If the cells start to multiply and grow and cause clinical problems like low blood counts or bone lesions, then we call it myeloma. And then some patients have very aggressive forms where the myeloma cells go out of the bone marrow into the bloodstream, and then it's really leukemia, plasma cell leukemia. And you can see, obviously, again, that's a little bit technical, but I hope you can appreciate that there are different clones of cells with different mutations and different aggressiveness, and depending how cells grow or what other influences they succumb to, essentially the disease can become also more aggressive over the years, and that's really what leads to treatment resistance and treatment failure. So what we did before I applied to the KL2 to have the preliminary data is we looked at expression data from healthy plasma cells and compared them to myeloma cells from patients with the most aggressive variant, which is the plasma cell leukemia. And there we could see that PHF19 was the most aberrant deregulated gene. So it was highly overexpressed in patients with primary plasma cell leukemia compared to healthy objects. So that was the first time we came across PHF19. Interestingly, it has been described in some cancers and also in embryogenesis, because it is a regulator of transcription. And how it does it, it regulates histone methylation. So here, I hope you can appreciate that the DNA, and it's kind of wrapped around histones so that it can be packed into the nucleus. Now, these histones can be modified also by methylation. So here, for example, this is histone 3. Depending on where and how they're methylated, they can turn on transcription, meaning that proteins are being, or RNA is being transcribed and then translated into proteins, and a lot of proteins are being produced. However, PHF19, what it does, it binds to these histones that curb on transcription, and then through various pathways, mostly the PCR2 complex, and then through these subenzymes, it actually turns off. It turns off transcription. So it's a kind of a silencer. And so with that data, and again, with also some manuscripts that had been published on PHF19 in cancer, as well as EZH1 and EZH2, which are subunits downstream of PHF19, I applied for the KL2, really looking at the aims being to see whether PHF19 expression is a prognostic marker in multiple myeloma, and then also what the biological function is. So what we could see from our patient database is that PHF19 expression highly correlates with disease aggressiveness. So you have normal plasma cells, and then plasma cells, or aberrant plasma cells from patients with these precursor disease stages, which are MGUS and smoldering. They have typically a lower level of PHF19 expression. And then once patients develop multiple myeloma or even primary plasma cell leukemia, the levels go up. That was also true for patients with relapsed multiple myeloma compared to newly diagnosed multiple myeloma. There was an increase of PHF19. And again, since relapsed multiple myeloma is usually associated with more resistant and more aggressive disease, we also concluded that this would mean PHF19 is really a marker of aggressiveness. And then we looked at PHF19 expression in different subgroups. So multiple myeloma can be divided into different subgroups depending also on different other expression of other genes. And what we could see is that PHF19 was higher in those groups that we know tend to be more aggressive and tend to have shorter survival, which is the T1114 group, here CD20 negative, as well as MAF and MMZ. And then survival analysis, just based on PHF19, equally showed that those that have a high PHF19 expression do much worse. So their survival is much worse than those compared to those who have low PHF19 expression. And then interestingly, we found a bioinformatician group that's located in Washington. Somehow they were also working on PHF19. They got to this gene through different analysis, and we ended up collaborating and really could confirm that PHF19 as a single gene is the best predictor of outcome in multiple myeloma. So after we then established that PHF19 has definite prognostic impact in multiple myeloma, we went on with biological studies, in vitro and in vivo. Meaning we really wanted to see what the function is of PHF19 and what it does in multiple myeloma cell lines. So we used two common myeloma cell lines called JJ3, ARP1, and then transduced them with a knockdown plasmid. So essentially what happens is that the plasmid you put into these cells can knock down PHF19 expression, as you can see here. So this is the control, and this is the knockdown, and the expression of PHF19 is then artificially lowered significantly. And that was true on a shRNA level, or on an RNA level, as well as on a protein level, as seen here with the Western blot. There was knockdown of PHF19. And then we did studies to kind of see what happens to growth, colony formation, and what would be the mechanisms that lead to any differences. So one thing we did was cell growth measured here by viability assay. So what you do is you take your JJ3 cell line, incubate the knockdown and the control, and then compare how many cells are still alive or proliferating after 24, 48, 72, and 96 hours. So here the ratio is essentially the knockdown versus the control, and you can see that compared to the control, there was a significant decrease in growth of in both cell lines. And that was true also for colony formation. So what you do in colony formation is you take one cell and plate it in kind of a semi-liquid gel into a small well, and then you wait 2 weeks and see if the cells grow or not. And if, in a normal state, these cells will grow, and actually from one cell, they will form a colony with this kind of huge bulk of cells. The knockdown, though, didn't really grow nicely and only formed a few or very small colonies. And this is just the same picture, but with red fluorescence, because, and I didn't mention that, but what we did is the plasmid also carried a red fluorescent protein, so we would actually be sure we would only see the transfused cells. So, and that was true for both cell lines in terms of growth. The ARP, colonies also were smaller overall, but these colonies were a little bit more widespread, so it's a little bit difficult to see. So we established that growth, proliferation, and colony formation was decreased in those with PHF19 knockdown compared to those who had normal PHF or high PHF19 expression. And then we wanted to see, well, what is the mechanism? Is it because the cells die, or is there a cell cycle, inhibition in cell cycle? So we did not see a lot of difference in apoptosis, at least not after 96 hours. But we did see that the cell cycle was that PHF19 knockdown significantly led to a decrease of myeloma cells within the active cell cycle. So you might remember that cells kind of go through a cell cycle and then eventually divide, and then again divide. So that's how they grow. And you can see here the purple line is the control. So this means that a lot of the cells are in the part of the cell cycle where they're actually close to where they're growing, because they're close to dividing. Whereas the red is the knockdown, and it shows that there's a shift towards stages of the cell cycle which are actually more dormant, and there's not a lot of growth. That was true for both cell lines, maybe more so for the JJ3 compared to the ARP1. But you can see here that, in both cell lines there was a decrease of myeloma cells within the active cell cycle. So yes, so we had by then a couple of results, meaning again, decreased proliferation, decreased cell cycle, decreased colony formation, and then we wanted to look at, well, what are the downstream targets of PHF19, or what pathways are altered by PHF19 knockdown. Again, so we did first Western blot, and really also wanted to make sure that knockdown of PHF19 truly is consistent with H3 with the decreased trimethylation of the histone, which was the proposed mechanism from other studies, but we hadn't really shown it. So here again, we show that the trimethylation of this particular histone was decreased, similar to EZH2 inhibitors. And again, this was one of the subunits that was regulated also by PHF19. And there are right now actually EZH2 and 1 inhibitors in clinical trials or commercially available, and that's how we were able to obtain those. And yeah, so similar to EZH inhibitors, we could see that PHF19 leads to a decrease in this trimethylation of this particular histone. And this then led to this is a pathway analysis. Essentially, it just shows what shows the genes that are differentially regulated by PHF19 knockdown. What pathways are they associated with? So we used bioinformatical program, and it shows that a lot of genes were really associated with cancer, meaning that they have been associated with tumorigenicity of cancer or oncogenesis. So that was also nice because it kind of confirmed our hypothesis that PHF19 is really important in multiple myeloma, but also in other cancers. And then when we looked not just at the pathways, but at the genes that were actually affected by PHF19 deregulation, we could also identify certain genes that have been really shown to be important in multiple myeloma, including EGR1, which is a gene when increased causes resistance to treatment, and it was actually downregulated in both cell lines after a PHF19 knockdown. Similarly, BCL2, which is in MYC, you might know this one, which are actually very known oncogenes in cancer biology, were also decreased. The expression was decreased after PHF19 knockdown. SLAMF7 was increased after PHF19 knockdown, and the reason I'm showing is because there actually is now a drug that targets SLAMF7, suggesting that if you could inhibit PHF19, which would increase SLAMF7 expression, the drug might actually be more potent. But that's quite hypothetical. So this is on an RNA level, and then we looked also at mass spectrometry, what proteins are actually affected. Again, there are some differences in proteins and RNA in the genes that we measured by RNA sequencing and then by mass spec. And this is mainly because these genes are expressed at so low levels. They're transcription factors, so the expression is so low that the proteins are just very difficult to pick up because the quantity is just very low. However, we even without actually seeing these genes or these proteins in the protein analysis, in the mass spec analysis, we did see alterations in proteins that have been described to be important in oncogenesis. So essentially, that all led to a conclusion that yes, PHF19 is plays a major role, at least in multiple myeloma, for tumor growth and tumor proliferation. And then the last part we did was also in vivo experiments. So we used not skip mice, implanted the cell line, in this case JJN3 cell line. 10 mice had PHF19 knockdown and 10 had a control. And so that was done all through tail whale injection. And then, yeah, they needed to start treatment with doxycycline. And I didn't mention that, but the shRNA knockdown model that we used required doxycycline to become active. Otherwise, the cells would just not, there would not be a knockdown if you wouldn't induce them with doxycycline. And then we measured the tumor markers, meaning light chains, which is a tumor marker for myeloma, by ELISA weekly and as well as bioluminescence by IBIS bioimaging studies. And what we could see is that the overall survival was decreased in the control cohort, almost statistically significant. And there was also there was an increase in the tumor markers in the mice that had the PHF19 knockdown plasmid. And we saw also similar results with the bioimaging. So you can see here, this is the control mice, this is the knockdown mice, and this is just exemplary, taken few mice to show how the growth is. And obviously the PHF19 knockdown didn't stop the growth completely, so the mice would still develop some tumors, but it was certainly delayed the growth. And this is the bioluminescence measured, the intensity measured. It was a huge variety, but overall the trend was there. So in conclusion, we saw that PHF19 expression is associated with disease aggressiveness in myeloma, and that high PHF19 expression correlates with adverse prognosis. We saw that PHF19 knockdown decreases the growth of myeloma cells in vivo and in vitro, and that PHF19 regulates major cancer players that in multiple myeloma, which are very well known, BCL2, MYC, EGR, and SLAMF7. So targeting PHF19 could be a new therapeutic avenue in multiple myeloma. Currently, there are no inhibitors available for PHF19. And then just, I think one of the last slides is what we did with the results, so publication, abstracts, and funding. So the project itself, meaning PHF19, led to 3 publications. One is still under revision, but more so also what the KL2 really provided me with is protected research time. So we were able to publish a lot on other projects also. So that was really very helpful to me as a clinician to have protected research time. Then a grant submission. So I started applying for grants in 2018. Small grants really, mainly on the PHF19, but I got good feedback. However, none of them got funded. So I'm still kind of wanting to proceed with the project, but right now the emphasis of the research has shifted a little bit because I got the chance to actually apply for another grant, one of the COBRI grants here at UAMS. That is the COBRI grant for studies of host response to cancer therapy, and there the topic is just very different. It's using therapy-induced damage to the host bone marrow, using single-cell RNA sequencing. So that's what I've been working on now most recently. But I hope that eventually I can also maybe bridge back to the PHF19 and expand this topic. Yeah. And with that, I want to thank everybody who supported me in the KL2 research, and I'm open for any questions.
Thank you so much, Dr. Shinky. Not seeing any questions, and we are like maybe a minute behind, so we will move right along to Dr. John and her project.
Hi, everyone. Okay, I'm going to share my screen. So hi, everyone. I am a clinical child psychologist and a researcher in the Psychiatric Research Institute. And I'm going to talk to you a little bit about my KL2 and how it really informs some skills within career development, as well as some knowledge from the research that's associated with some of the current research that I do. So broadly speaking, I'm going to introduce a little bit of my background that informs the work that I do. So as I said, I'm a licensed child psychologist. I'm an assistant professor, though excitedly an associate professor starting in July in the Department of Psychiatry. So my clinical specialization focuses on early childhood mental health, especially in infancy, toddlers, and preschoolers. So 0 to 5-year-olds are totally my jam. I am certified to provide and train several evidence-based trauma treatments. And I really focus my work on T3 type of translational science, which really focuses on translation of best practices into clinical practice settings, where I use implementation science to try to figure out how do we infuse trauma-informed clinical best practices into all the settings in which young children are attending. So my KL2 research focused on young children with behavior problems, which is present in 1 in 5 children nationally. And it's actually the second most common reason that children are referred to mental health services if they're under the age of 5. And behavioral difficulties are exceptionally important to pay attention to because especially when left untreated, they can predict significant adolescent and adult difficult behaviors. Things like adolescent truancy, delinquency, as well as adult criminality, violence, substance use. And so there's a lot of focus on identifying risk trajectories, and behavioral problems certainly have a large risk trajectory for young children. And so traditionally, what we do in mental health is realize that in order to change young child behavior in this trajectory, you're really needing to focus on the caregivers. So we thought that's parents, of course, and that's who we're going to bring into treatment and do this work with. However, now the landscape of caregiving has been dramatically changing in our country, such that 80% of children, including my own, spend a majority of time in non-parental childcare. And so our treatments that focus on changing parents are missing actually the bulk of caregiving that's happening for young children. So at the same time that children are receiving these outpatient mental health treatments from clinicians in clinics like the UAMS Child Study Center, they're also separately receiving treatments that work with their teachers to change their behaviors. And so an ideal clinical best practice is for that therapist who is working with teachers and that therapist who is working with parents to coordinate their care, to make sure that kids are getting comparable things across environments. But studies show that these services are actually rarely coordinated, but it's unclear why. So what are the barriers and facilitators to promoting coordinated care in early childhood mental health? And so I was able to get a lot of foundational training in qualitative methodology through my KL2, which is something I had not yet had the experience to do. We did a lot of targeted focus groups where you can see that we were able to get almost every single provider in the state at that time who was doing school-based work into a focus group. And I was able to get a lot of training. Thank you, Tracy Abraham, for helping me with template analysis to really focus on extracting the data in a meaningful way. There were 2 portions of this data, but I'm going to focus you on the area in blue because I think it most relates to the current research that I'm doing now, where it's really talking about like, how do we identify facilitators and barriers between different settings in which clinical practice is not happening, which you'll note is going to be a common theme as we proceed into my research now. So template analysis for this group demonstrated some of the things we expected to find. That like one of the things that's hard about coordinating care is like, who has the time to do that? So pragmatic barriers were something we kind of expected. But there were some more relationship, social barriers, and theoretical barriers that were interesting. So it wasn't just that I don't have the time to coordinate. It also might be that I don't know if I want to coordinate with these people because we don't approach things in the same way. I don't feel respected by those providers. And that brings us to an interesting point of implementation science and the degree to which relationships matter when we think about implementing best practices. And so out of that research in my KL2 was the emphasis that implementation science can't just focus on the pragmatics, the things that we think are getting in the way for people, because there's actually a lot of deeper theoretical and interpersonal difficulties that make this a challenge. And can we, as scholars, help to make these relationships that promote best practices? So currently, here's the types of things that I am working on. So it's a great position to be in right after the KL2, literally the day after our KL2 award period ended, I started our next project. So this is a grant that is funded through the Victims of Crime Act. It's basically dollars that are collected from offenders for various treatment types of programs that they need to do, are pooled together and actually fund services and research for victims of crime. And this is federal dollars that run through state funding. And the Voca Grant is twofold. The first being that this Voca Grant, I should say, focuses on bringing best practices into child welfare. And child welfare is foster care, otherwise known as. And what we know is foster care is a setting that is certainly interacting with vulnerable children with mental health needs, and is also a setting in which trauma-informed best practices have trouble holding on. And so what the Voca Grant has done is allowed us to expand and pilot services to improve coordinated care and implementation of best practices in child welfare. So the first is that some kids need more comprehensive evaluations that are high risk in the child welfare system to make sure their diagnoses are right and where they're going and what treatments they're getting are important. And so since October 2019, we've been able to triple the capacity for the amount of evaluations that we're able to do for DCFS each year through expanding this clinical service outside of UAMS by training community psychologists across the state. We're also piloting a coordinated care model in which a social worker pairs up with a DCFS caseworker to actually get people into the treatments that we know work. Mental health care is a lot like any other service where some mental health care is targeting some things and not all mental health care is created equal. And what we know is that kids who've experienced trauma need to be in treatments that have shown to work for trauma. And so what this program does is piloting a coordinated care model in which caseworkers build relationships with a specific social worker that will help them then guide their families into treatment. Because remember from the KL2, it was really clear that it wasn't just the pragmatics. Caseworkers didn't just need someone to help with time management. They needed relationship building. They needed to trust the person that they were insuring their kids to. And so a large part of this coordinated care model is building that relationship over time. It's served 107 children so far, or rather I should say 107 families so far since October of 2019 with really promising results. What we're seeing from caseworkers who are surveyed is a vast majority of them are increasing in their knowledge of best practices and their understanding for the need of mental health practices, learning themselves what constitutes best practices in trauma, and really openly and qualitative feedback discussing that the relationship they're forming with that social worker helps them to provide the best clinical care and practices that they can for the children that they serve. So the other grant that I co-direct is continuously funded through the state legislature. It's called the Arkansas Building Effective Services for Trauma, Our Best program. So we are legislatively funded each year with the broad mission of improving outcomes for children and families who have experienced trauma. And we do that through a multitude of ways. But the ones that I think are most relevant to this conversation is really providing wide-scale dissemination and implementation of best practices in the community and implementation support to specific settings in which children are having their mental health care needs taken care of. And so I'm going to talk to you a little bit about those outcomes. I want to be really clear here, though. The Our Best program has been in existence since 2009, and I took over directorship in 2018. So when I present you with some of this data, I want to just be super clear. I am by no means taking ownership for this level of progress. Our Best is a really highly functioning team. And I want to pay tribute to that team and make sure that that's really clear. So how has implementation science helped us to change the landscape in Arkansas? So since 2019, over 47,000 children have been served using trauma treatments that they were trained from our program. So clinicians who we train have served 47,000 children since 2009. What we've seen is a 63% reduction in wait times for children to get into appropriate mental health services. And 49%, so half of children receiving treatment, are within the normal range of trauma symptoms by their first follow-up evaluation. So not only are a large number of children being served, they're not waiting as long to be served, and they're getting better quicker, such that we would expect with evidence-based treatment. So anybody who's a disseminator knows that we can train people all we want, but if they don't use the treatment well, what good did that training do? So it's important to pay attention to outcomes as well as just numbers. And so this is really exciting to see those outcomes improving dramatically. When I say wide-scale dissemination, I mean it. So in the past, since 2009, we've trained about 2,500 therapists across the state of Arkansas. All of this training is free to them because it is funded legislatively. And we have now trained therapists in 66 of 75 counties in Arkansas with evidence-based treatments that start at birth and go all the way through 18. And so finally, what we can see if we look at a special practice setting called child advocacy centers, which are a one-stop shop for children after abuse, particularly sexual abuse. We provide them with a lot of implementation support and training. And they've had an 8-fold increase in the amount of children that they're able to support using the funding and training we provide for them to do that. So the last thing I want to talk about is a little bit about future directions. So I want to pause here for a moment just to slow down a bit. That my type of research, translational research in the T3 category, is a lot different than basic sciences research. And so one of the things I tell people a lot as they're getting into this process of trying to figure out how to get funded is to think about diversifying funding. So I have applied for many NIH grants and gotten none of them, but have been continuously funded since the KL2 on non-NIH funding. And so as you're thinking about the types of funding streams that could best promote your type of research, one thing I have found, at least in my experience, is in these T3 types of scholarly work in which the focus is really building capacity and promoting translational research into practice, I've just found a lot more success in funding those things outside of NIH into more, into just different funding streams. So overall, I would just tell people, like, go where the money is for the specific type of work that you are doing. And at least I found in T3 work, state funding has really helped us launch a lot of the projects that I direct. So in terms of future directions, the first thing we're going to be doing in 3 weeks, is submitting for a second round of Voca funding to again expand the number of psychologists trained in complex trauma assessments, and again expand the amount of counties we're serving in trauma care coordination models. So we'll bring on another social worker, pair it with caseworkers from another county and see, does this replicate? Because we've only tried this in Pulaski County, and Pulaski County is unique in the state of Arkansas. It's where Little Rock is. It's a very urban setting. And so the next round will be piloting trauma care coordination in a rural county to figure out, does it work the same way? And that's really that translational idea of how do you get best clinical practices working the best for the specific setting that you're in. And so we're going to try our best to replicate the great findings we have in an urban setting into a rural one with the second round of Voca funding. And the next is we recently applied for a SAMHSA grant that focuses on supporting wide-scale dissemination of trauma-informed schools. So this goes back to, another lesson I've gotten from the KL2 is keep your research doors open. So I've now been away from the school sector for 2 years after my KL2, doing work more in the practice sector. And now we're bringing it back to schools again. So this project supports wide-scale dissemination of an organizational change model for schools. Basically, what that means is if you want to implement trauma-informed practices, that takes system-wide change. It takes training your administrators, your teachers. It takes forming crisis plans that are trauma-informed, referral streams for best practices, staff wellness to ensure that they're able to provide what they need to. And so organizational change models are an implementation beast, as you can imagine, because there's so many moving parts of what you're trying to implement. So we have now successfully piloted a 2-year organizational change model in 2 school districts in Arkansas, in Russellville and North Little Rock. And we are applying for funding to try to support wide-scale dissemination of that model nationally. So in the grant, we would be testing that model in 2 different states in the country to see, again, does it work outside of Arkansas and what changes need to be made for a national setting? And hopefully being able to get the funding to support that project outside of our state. And so I just want to make sure to say thank yous to so many people. I have so much gratitude for the KL2 and the skills it gave me, the relationships it gave me. It was truly invaluable. Thank you so much to former and current KL2 directors. Thank you, Nia. Thank you, my fellow KL2 scholars. It's nice to see you guys. And so thanks so much to the Our Best team, especially my co-director and program manager. And then finally, thank you so much to the countless adults who fight tirelessly on behalf of vulnerable children in our state. There are too many to name, but this T3 research has really shown me how difficult and rewarding it is to bring best practices into routine settings. So I'll stop there, hopefully saving Lisa enough time to get through what she wants to get through as well. But thank you so much for your attention.
Thank you so much, Dr. John, and we will move right along to Dr. Brentz.
Okay, changing gears a little bit to pharmacology and development of a new, hopefully improved pharmacological agent to help with neonatal opioid withdrawal syndrome. So particularly that associated with buprenorphine treatment during pregnancy. So buprenorphine is a partial agonist of mu-opioid receptors. It's used to treat opioid use disorder, and one of its adverse effects is dependence and withdrawal, like pretty much like any other opioid. So buprenorphine is used during pregnancy in women who have an opioid use disorder, and we've seen in the last several years that abuse of opioids during pregnancy has drastically increased. You can see in 2004, the incidence per 1,000 births was 2, and by 2014, it had more than quadrupled for all payers to 8. So
Neonatal opioid withdrawal syndrome is makes in short version, it makes children very sick, and they often require admission into neonatal intensive care units. And this is also very costly, so we see the cost associated with neonatal opioid withdrawal syndrome drastically increase as well. So let's see. Trying to figure out how to move the little pictures, the video here, so that I can actually see my slides. Okay, I'll just move that out of the way. So currently, if a woman with opioid use disorder becomes pregnant, seeking treatment and obtaining medication-assisted therapy is currently the best option for her. However, one of the side effects, of course, is dependence and withdrawal, and that's true in an adult human, that's true in rodent models, like I'll show later on. That's also true in fetuses. So whenever they're born, they end up going through withdrawal, and that's what NALS is. So the question I'm trying to investigate is, how can we improve treatment using buprenorphine during pregnancy? And there's one thing that's very important to know about buprenorphine is that there's, interestingly, no dose-dependent relationship between mom taking the buprenorphine that mom takes and the withdrawal that her newborn goes through. Presumably, just because of the way we know pharmacology works, exposure in utero should be proportional to dependence and withdrawal. So that leaves open the question of what happens between mom taking the medicine and the fetus being exposed. And that leads us to consider pharmacokinetics, which in short is absorption, distribution, metabolism, and excretion of the drug. So my work has focused on better understanding the distribution and metabolism of buprenorphine and how that affects the fetus and ultimately the newborn. One thing to know about buprenorphine is that it has an active metabolite called norbuprenorphine. I'll call norbup from here on out. So it is formed whenever buprenorphine, this group on buprenorphine is cleaved by cytochromes P450s to form this structure. Some things that we know about it is that it, like its parent drug, has very high affinity for the opioid receptors. Unlike its parent drug, it has full agonist efficacy at the mu opioid receptor. It's also known that in pregnant women who are taking buprenorphine, the concentrations in their blood of buprenorphine and norbuprenorphine are roughly equal, so that suggests that norbuprenorphine may be able to compete with buprenorphine at different target sites. Another study correlated the umbilical cord blood concentrations of norbuprenorphine at delivery with the severity that the child eventually, severity of withdrawals that the child eventually goes through during the first week of life. Interestingly, buprenorphine concentrations were not related to NAS severity. So that tends to point to norbuprenorphine having some sort of a role in dependence and withdrawal of the newborn shortly after delivery. In our lab, we developed a rat model of neonatal opioid withdrawal syndrome, and we used it to test whether or not norbup by itself can induce withdrawal. So you can see we administered to pregnant rats one of 3 doses of norbup or morphine or vehicle, morphine being a positive control. And we challenged the newborn pups within 12 hours of delivery with an antagonist, naltrexone. So in an opioid-dependent individual, giving them an antagonist of opioid receptors will precipitate withdrawals. And then we measured using a scoring here. We're able to measure withdrawal in the pups. And you can see that norbuprenorphine elicits withdrawal on par with a good solid dose of morphine. So that suggests to us that norbupe can cause NALS. So our overall hypothesis that we're investigating and are testing in my lab is that norbupe contributes to bupe-associated NALS. And why is this important? Well, if we could somehow decrease exposure, fetal exposure to norbupe, perhaps we can decrease the adverse effects that it has on the fetus. So a couple of strategies that I've already foreshadowed. You can decrease distribution of this metabolite to the fetus, or altogether decrease its formation. For today, and I've been investigating both of these potential strategies, but today, because it's what I've pursued in the KL2 project, as well as a follow-up R21 project to further build this project up, I'm going to be talking about a way that we're trying to decrease norbupe formation. So the overall strategy, this is a diagram illustrating the maternal fetal unit, mom's brain, fetus, and then here's mom's blood with approximately equivalent concentrations of bupe and norbupe here. So both of these readily cross the placenta, cross the immature blood-brain barrier in the fetus, and distribute to the brain, where they can cause different effects. Buprenorphine readily goes into mom's brain. Mom's brain is actually the primary target site for therapy. However, norbupe doesn't
easily cross the maternal blood-brain barrier. And because that's the primary site of therapeutic action, that suggests that norbupe is maybe not required to successfully treat mom as long as you have buprenorphine on board. So if we can decrease that or eliminate it, that's less total exposure that the fetus gets to opioids. How to do that though, while preserving the therapeutic function of buprenorphine. So to figure this out, I went to Dr. Peter Crooks in the Department of Pharmaceutical Sciences and asked, is there a way, he's a medicinal chemist, I asked, is there a way that we can alter this structure to prevent this metabolism without drastically affecting its activity? And so one thing that he knew about this, the metabolism, is that the removal of these hydrogens is a critical step in cleaving this entire group. But if you switch out hydrogens for deuterium, which is the heavy isotope of hydrogen, that strengthens these bonds and is thought to make the structure more resistant to cleavage by these CYPs. So the idea is to make that change and hopefully decrease norbupe formation. So the hypothesis that we put forward is that D2 resists metabolism to norbupe but retains activity. And first thing we looked at is metabolism. Strategies for testing metabolism include using in vitro test tube assays where we have isolated enzymes, either the individual CYPs or liver microsomes, which contain a plethora of the different enzymes, metabolic enzymes expressed in human liver. For a whole organism model, we also use a rat model. And just the basic idea is you have a system, you include drug, you wait some time, and then you measure your metabolite or your analyte of interest, the parent drugs. And for our in vivo, we also looked at other alternative pathways of metabolism to see if there was shunting to these other pathways like we would like to see. So we looked at glucuronides, which are also major metabolites. And we looked at those using LC tandem mass spec. So for this talk, I'm focusing only on my in vivo data. So for this, we gave IV televein injections to rats, and then, like I said, we quantified metabolites and the parent drugs. So what we found with our lower dose, what we were hoping to see, so in these panels on the x-axis, you have the time after the injection was given. On the y-axis, you have the analyte of interest. What we wanted to see was less norbupe in the animals that got bup D2. And we see a little bit of a difference, but not near as much as I would have liked. The parent drug concentrations were right on top of each other, suggesting that we gave the same dose and that they're behaving well. The glucuronides that are made from the parent drugs are also on top of each other. But what's really interesting is that the glucuronide of norbuprenorphine, there appears to be a difference. There's less of it in animals that got bup D2. One way to summarize this entire curve is using area under the curve. So when we look at area under the curve, we do see that there's a significant drop in the norbupe glucuronide in the animals that got bup D2. So I wasn't really expecting to see that with the glucuronide, but I think it's very promising. We look at norbupe, there appears to be a slight drop, but not statistically significant. So what does this mean? Where does the glucuronide come from? So it comes from norbuprenorphine is one route. So perhaps there is less norbupe present from in the animals that got bup D2 to eventually be metabolized into glucuronide. And that's why we're seeing this difference. Another potential explanation is that after the parent is glucuronidated, it could still be cleaved to produce norbuprenorphine, but the bup D2 glucuronide also resists this cleavage. So I thought that if we move up a dose, maybe we'll see more of a dynamic range and more of a separation between our two groups. What's really interesting, of course, things never go like you expect them to go, right? We actually saw a complete disappearance of that difference with the glucuronide as well, and a very, very tight data here with norbuprenorphine. Very concerning is that there seemed to be no difference, even though we increased our dose by 2.5-fold, there seemed to be no difference initially in the metabolites in the two different dosing groups. So you would expect that you'd have more metabolite in this group. We did see an increase in our parent drug and the parent drug glucuronide, suggesting that we actually did increase our dose like we were expecting to, or we intended to. And we also analyzed the formulations, and they were the concentrations we wanted them to be. So this appears some sort of saturation or ceiling effect with the metabolites with these pathways of metabolism at this pretty high dose right here. So one of the things, if you remember, I said about humans is that roughly norbupe and bup plasma concentrations are roughly the same in humans that are chronically taking buprenorphine. And one of the things that's a little bit difficult about this is our rat model doesn't exactly replicate that. So you can see when you compare the parent drug concentrations with the norbupe concentration that there's a much bigger difference. So that's a challenge that I've really, I'm still working on exactly how to overcome that to help with testing my hypotheses that I have about norbuprenorphine and buprenorphine during pregnancy. So one of the things that I want to see is that the bup and bup D2 have the same pharmacodynamic activity. That is that they bind to the receptors and they activate the receptors the same way and have similar effects. So one way that, or the way that we looked at Affinity it's a competition binding assay. We have a drug that we know binds to our receptors of interest, and then we're going to measure how well those are able to, how well that drug, and that drug is radio labeled so we can quantify it. So I'll go through this quickly. We incubate, terminate the reaction, and count it using scintillation counter. So what we expect to see as we go from low to high concentrations of our unlabeled drug is that our labeled drug, which is normalized at 100% here at the top of the curve, will binding of it to the receptor will decrease. That's what we see with morphine as our positive control. Bup, much higher affinity than morphine as we expected. Bup D2, what we want on top of buprenorphine, and it is. So that was really good. So the other opioid receptors, we see pretty much the same thing. The kappa pretty much on top of each other. So that's good. It still retains the affinity of the same affinity as buprenorphine for the opioid receptors. So here's a measure of affinity taken from those curves. And what this means is that bup and bup D2 have the same affinity, and it's greater than morphine affinity for all three of the opioid receptor types. So downstream of the receptor is G protein, which mediates the effects of the activated G protein coupled receptor. So how this works is that you have a G protein that's inactive. If it's inactive, it's bound to a GDP, diphosphate guanosine. When it's activated, when the receptor becomes activated, that switches the GDP to GTP on the G protein. And then whenever that's hydrolyzed, it returns the G protein to the inactive state. So the way this assay works is that we have a non-hydrolyzable analog GTP that's radioactive, so we can quantify it. But once this goes on to an activated G protein, it doesn't come off. So we can quantify how the receptor and the drug binding to each other activates G proteins. Similarly, this is a test tube assay where we have our GTP analog, our source of receptors, GDP to shift them all into an inactive state to begin with, and then varying concentrations of a full agonist positive control, our test ligands. Same thing, we let it incubate and then we filter. So what we see here is that at H more and H core, our positive controls work very nicely. We see a nice activation with increasing concentrations of our full agonist. Bup acts as a partial agonist, like we would expect. It doesn't fully activate, but does partially. And bup D2 does the same thing with approximately the same potency and efficacy at both the mu and the kappa opioid receptor. So bup is known to not really activate delta. So we did just a quick screen with a really high concentration just to see if we can get any activation. And as we expected, we did not. Same thing with bup D2. So conclusions, pharmacodynamics, so equal affinity, equal potency and efficacy at the opioid receptors. That's good. It suggests that it's going to still act like bup acts. And I didn't show these data, but we did a quick and dirty analgesia assay and showed that again, the bup D2 curve is right on top of the bup curve, like we would like to see. So with pharmacokinetics, is a little bit less or a little bit more ambiguous. I didn't show the in vitro data, but we see about a 15, maybe 20% decrease in formation of the metabolite norbup. Would have liked to have seen more. So I'm still trying to work out exactly what the next step is as far as like what the metabolism of BUP-D2 looks like relative to metabolism of BUP. So, and then like I mentioned before, the model differs from humans. So that makes it a little bit difficult to predict how it would work in humans. Things to investigate, okay, humans are taking buprenorphine chronically, so do we still see similar pharmacodynamics when BUP-D2 is given chronically? PK characterization during pregnancy, I've tried that. It's a long story. Again, the issues with the model and the differences in metabolism have kind of tripped us up, but let's see. So one of the initial thoughts whenever I was formulating this overall hypothesis that norbup is possibly affecting neonates, fetuses, neonates, is that it's difficult to test BUP and norbup interactions in our rat model because BUP is metabolized in norbup. So you don't know how much of that norbup comes from the exogenously administered norbup versus that it's metabolized. Now that we see that that's not really so much an issue, we can go on and do those interaction studies. And then further characterizing BUP-D2 in different models of treatment to make sure that it is still an opioid appropriate treatment for opioid use disorder after it's deuterated. So just want to acknowledge my collaborators on the project and on the R21, as well as the KL2 project, KL2 mentoring team, students that have helped and worked on this, and technicians that have worked on this project, as well as my department and department chair and funding.
Thank you so much, Dr. Brents, and thank you, everyone. And I hope you have a great evening.